I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them. Hey, so we talked about this in a previous episode. Do you know what are digital twins?
No, I've never heard of the digital twin.
Can you go to work for me?
I have no idea what a digital twin is.
Is that when you have an avatar that looks like you?
It's a way of mirroring like an actual process, like in an artificial sense.
Digital twin, like when you just collect a lot of data on like a person and they kind of try to simulate it.
And follow up, if you could make a digital twin to test some hypothesis, what would it be?
If I had a digital twin, I feel like I would just try all these different little drugs that seem to have magical effects on people. Like, GLP-1 receptor agonists. I would be curious. Like, I don't know if I love eating. So, I wouldn't want to take them, but I would be curious what the kind of effects they would have on me.
Hi everyone, welcome to the show. I'm Maria Wilson. So on today's episode, we're picking up a thread of a conversation from season 5 that we all found super interesting, digital twins. So I'm here with Iraj Hosseini, who works to develop digital twins. Welcome to the show, Iraj.
Pleasure to be here. Thanks for having me.
Thank you for coming. So last season, we had an episode which we called Merging Minds and Machines, the Future of Drug Discovery, and we interviewed a computational biologist, John Marioni. And he brought up this concept that could be very interesting for drug discovery and development called digital twins. And I think he also used the term adaptive experimental design, like adding things to models as we go along. And we thought this was really, really interesting and deserved its own show. So we wanted to have you here to talk about all of these things to do with digital twins. And could we start by you just telling us a little bit about the concept as it applies to both medical research or even more broadly?
Absolutely. A digital twin of a patient or an individual is basically a virtual mathematical model that encapsulates all the data that you have for that individual or patient. It could be physiological data, it could be measurements that you have from the lab work, it could be like weight and height, it could be genomic information, it could be the type of treatments that they are using on a daily basis. All of that incorporated into a mathematical model is what we mean when we say digital twin. Now, there are things that you measure and you can directly incorporate into your computational models. Like, for example, if someone is taking a medicine, I can measure the concentration of that drug in their system, and then I can directly put that in the model. But there are things that you don't know. For example, like if they have T cells and if the T cells are being activated, you don't necessarily know the rate of activation. And that's, I guess, both the kind of like a feature and a bug for digital twins. It gives you a lot of power because you can incorporate that uncertainty into building it. So you only have one patient, but you can create thousands and thousands of digital twins and say, what if? Like, what if this person has this rate of T cell activation? Or what if this person has this particular treatment that they are taking in combination with this drug? So that's the power, but at the same time, it gives you ranges. So we cannot predict exactly how a patient responds, but we can say this could be the range of responses that you see for a particular individual or a patient.
How do you decide what data is relevant to build the twin? Like, for example, you could put into your model that I've got 5 tattoos and I dye my hair blonde, but is that relevant to what we're talking about?
That's a great question. I guess the way that we are looking at it is like we define our digital twins in the context of a clinical trial, typically. Like, for example, if you have a clinical trial targeting cancer patients in heme indications. So those are the type of things that like you kind of think of, like maybe it's important to include the lymphocyte numbers for this patient. Maybe it's important to look at whether these patients have taken other treatments that are relevant to their cancer, and those are the type of things that you include. But for example, you would not go and say, I need to know like the height of this patient. That may have little information relevant to what you are building.
And are there circumstances where you would build in more potentially irrelevant information to do signal seeking? I mean, could you ask questions about, for example, is height relevant? Is age relevant? I mean, are the models made more powerful by having more information in, or does that make them more confusing?
I guess it's kind of like a yes and no question. Sometimes we look at like the previous clinical trials that we've done, and if there are things that are relevant and we've basically identified that these things are correlated with the outcomes that you have, then those are of more interest. But also you want to have causation in the parameters that you're incorporating into your system. You want to know that it's actually relevant with the mechanism of action that you know of that drug. So all the in vitro data that you've done, like you've looked at basically a whole bunch of in vitro assays to understand the potency of the drug, the actual mechanism of action of that drug. You also know about the modality, like whether this is an ADC, antibody drug conjugate, is it a bispecific, is it like a T-cell engager? You want to make sure that the models are informative, but also you want to make sure that you can communicate the results, right? Therefore, there is a limit of like, if you make it too complex, then it might be hard to sort of like interpret the results. So there's a trade-off.
Hi, Maria.
Hello, Wellington.
Hi, Maria.
Hi, Karen.
So, clarification question. I often hear in these conversations that we've had the term signal seeking. And I get that. I get that we're looking for something or some information pops up, but I wanted to ask you, what is it exactly?
The term is used in clinical trials when you're running an early stage clinical trial, maybe the first time you're in patients, and you're just looking for some evidence that a drug is engaging the target, that it's modulating some biomarker, that it's moving, some endpoint. So we call that a signal seeking study because it's not like one specific defined endpoint or outcome that you're looking for. And if you get a positive signal seeking in a signal seeking study, then you might give you confidence to move forward to do a more rigorous, say, placebo-controlled study. And what you're doing when you're changing one variable at a time, is that so you might change or the dose of the drug or the baseline level of T cells or something, and you can kind of look at that. Is that kind of what we're doing?
Correct. So that would be the first level of like analysis that we do, like single variable type of a thing. Is like, so I'll give you like an example of like a real case that we've done. So typically when you run a clinical trial, you have 40, 50 patients. They have received different dosing schedule, different dose levels. Let's say they all have NHL or non-Hodgkin's lymphoma, but they have different subtypes of that disease, right? So you really have a very sparse, diverse data. So what we do is like we take all of those patients and we say, let's create 1,000 digital twin for each of them. So if my clinical trial is like just 50, I'm actually creating 50,000 virtual patients. Yeah. And each patient in reality just experienced one experimental condition, right? So they received one dosing schedule of that treatment, and I have the observation for that. But that particular patient was not exposed to other dosing schedule, other dose levels, or that patient didn't have other subtypes of the disease, right? So what we do is like now we incorporate them together and say, these other like 49,000 patients, what if they were exposed to these different experiments? So basically you can think of like, there's a map, it's very sparse, but when you put them together, you can actually shed light on like different aspects of it and answer this question of what would happen if these patients had received something else. So that's the way that we look at it. But then we start with like single parameters and single variables and say, what if this drug was slightly more potent? What if I tried a different dosing schedule? But I feel like that's the beginning. Like you can do a lot of extension and say, what if I combine this drug with this other drug? What if I only look at the subpopulation of patients that have these many T cells and the tumor size is below this and the rate of doubling is above that? So now you can answer actually way more complicated and interesting questions.
That's so interesting because when you're designing a clinical trial, you've set your inclusion criteria. We spend a lot of time usually thinking about exactly who we want to have in our clinical trials, and we balance that with making sure that you're actually able to enroll the trial, right? But this gives you a lot more power from those that one or two individuals who signed up for the trial, that data that they're generating becomes infinitely much more useful when you can use it in models like this.
And absolutely, you hit it on the nail here, because essentially, I guess the next, because right now, our digital twins and the computational approaches that we use are very powerful. So, like, if you tell me, like, I need 1 million, I can create that very quickly. The challenge that we have is, how would you go from a patient observation and measurements to the actual system parameters that we need to have for that patient to basically guess what the rate of activation of the T cell would be, right? And I think that's the golden opportunity, that as we basically gather more genomic information, single cell RNA-seq, we might be able to just go from those measurements and map them to the system parameters that we have in the model. So therefore, when you give me a new kind of participant in a clinical trial, I would look at the measurements, the genomic information, and other information, and say, these are the system parameters that I have, or I guess I would have for this patient. So I would be able to actually narrow the range of prediction for that patient even before treatment. So I think that's the way that we have to go, mapping the measurements to the actual system parameters, and there's a lot of work happening essentially in that space.
Yeah. It's obviously really, the data that goes into the system is obviously very, very important that we get that.
Absolutely.
Right.
And I think with like the cost of some of these things going down, with the fact that we are measuring a lot more data, as you said, like, we're going to understand what are the more relevant pieces of the data, and then we'll incorporate those into the model. Obviously, it's a little bit different from like the Gen AI that you have like in the entire internet at your basically disposal. Here, you only deal with like 50, 100 patients per clinical trial. So it's important to understand basically the totality of the system, like all the NHL patients, for example, that I gave the example, that have received different treatments. Can we learn from all of them, not just a particular trial that we have in front of us?
Currently, we're limited to our sort of using clinical trial data, but your hope is that we can get access more real-world patient data.
Absolutely, yes,
yeah.
yes.
So Maria, Iraj was talking about using genomic data in digital twins. What types of genomic data is he talking about?
I mean, really just whole genome sequencing, right? I mean, you can associate a response with a person's genetics. So, you might look at a polygenic risk score for diabetes or dyslipidemia or something and see if patients who had the polygenic risk score were more or less likely to respond to your drug.
Maria, I found myself thinking about our previous episodes about obesity, and with the possibilities that you're bringing up, it seems like there's many customizable simulations that we could do, and I wanted to ask, what would happen, or how would we think about digital twins in GLPs?
That's such a great question. So I think there's a lot of variables of patients, for example. I mean, obvious things like gender, like background medications, ethnic background, genetic heritage, all sorts of things that you could put in. I mean, one of the things we struggle with the GLP-1s is the tolerability. So trying to find a sort of digital twin models of what types of biomarkers or genetics or history associates with tolerating or not tolerating the medicines, for example. But yeah, the typical types of things that you evaluate when you're including people in a clinical trial, the BMI range, the with or without type 2 diabetes, all of these things, these are the parameters that you would create your digital patients using. Could be very useful. As a result of using digital twins, do you see any particular big breakthroughs already, or do you anticipate anything coming?
I think we have made a lot of progress on understanding the impact of the treatment on the patients that we have at the clinical trial. And that's very important, because the way that we look at it is we basically have one snapshot of a particular individual. They enroll into the clinical trial, we start measuring things, and from there to the point that we have an outcome, we can model that. And that is very powerful. You can do a lot of things with it. But the challenging part is like you don't necessarily know the trajectory of that patient, right? So you don't know how that patient would have progressed if there was no treatment. So when we are predicting things, you're predicting two things at the same time: the trajectory of that patient and the treatment effect. There's a lot of work happening on the AI aspects of things that we're looking at the population as a whole for a specific indication or a cancer and say, this is the trajectory of this population, and if you have some measurements, you may be able to actually predict with more accuracy of the trajectory of that particular patient if there was no treatment.
It sounds as if you're talking about modeling placebo, actually. You work deeply in the oncology space where using placebo is not okay, right? Nobody's going to sign up for, and it's not ethical to sign up for a clinical trial where you're not treated. And compared to some of the other diseases where you actually still can use placebo, it makes it pretty challenging in that space.
So as you said, like in onc, there is no placebo. So it's basically what would be the trajectory of untreated patients. But yes, in other indications, that would be exactly the thing. And I guess kind of like what makes it more powerful is like at this point, we have more confidence in predicting the response at a population level. Like we can say, if you have 100 patients, this would be the rate of outcome that you would be able to see, like 40% response. I guess we want to go to a goal of being able to predict at the individual level. And that's why we need a lot more data. You need a combination of these different. But I guess that's where the field is, right? A lot of development of methods, data needs to be collected. But once we are at a stage that we have enough data, these methods are, I guess, mature enough that can be combined and give you like more power into predicting individual outcomes.
How do you see all of this work meeting future patient unmet needs, especially in these, say, non-Hodgkin lymphoma and some of these indications you're really familiar with?
I guess the way that I think of it is like, what are the applications of digital twins? Basically, like think of just a normal clinical trial, right? There are a lot of different ways that you can dose a drug. There are a lot of different ways that you can schedule. Like you can say, I'm gonna go weekly at the beginning and maybe like monthly at the end. Or maybe you have some safety concerns and you wanna basically decouple your safety concerns from your efficacy. Maybe your safety is more acute, something that happens very quickly at the beginning, whereas your efficacy is something that you are seeing like it's a long-term objective. So that would give you basically a space to optimize, right? So, but practically you cannot do that in patients. You don't have, first of all, you don't have that many number of patients. It takes forever to look at all the different scenarios. So you use these digital twins and you basically run in silico clinical trials. What you can do is just run a bunch of different computer simulations. You look at a whole number of things and say, when I look at the heat map of all the possible scenarios that you are considering, this is the point that you need to be. And I feel like that's a very practical aspect of digital twins. It's the unmet need that you mentioned. For a lot of clinical trials, that's what we need to do. We need to optimize our dosing scenarios, and digital twins are going to address.
I really like that because when you think about just real-world patients dealing with a chronic disease, whether you have to go to the infusion center once every 4 weeks or 8 weeks or 6 weeks makes a huge difference to your life. And to your point, the way that has been defined in the past has been somewhat crude. I wouldn't say it's arbitrary, but like you said, you can't test 1 week and 1.5 weeks and 5.5 weeks. And so, you might ask as a patient, why do I have to go for this medicine every 6 weeks? And it's like, well, because of the 5 scenarios we tested, this was the one that looked best. Is there a better scenario? Maybe. And what you're saying is that you can identify that maybe.
And the way to think of it is like already it takes like 8 to 12 years to go from like drug discovery to approval if you have a successful drug. Our goal is to get these drugs to patients faster, right? So even if you can sort of say, I'm going to basically find a bunch of these scenarios and instead of testing 5 of them, just test 2 of them. That would save you like 2 years and you can get the drug to patients much faster. And that's the goal, right? At the end of the day, that's what we want to achieve. We want to make sure that the patients that need these drugs, they can get them faster.
So it sounds like we're obviously not going to be able to completely replace clinical trials, but we'll be able to get more data out of less patients and less effort to help us drive decisions about whether drugs should be advanced and what doses we should use.
Absolutely. The other unmet need is because of the diverse set of patients in clinical trials, sometimes finding a biomarker would be difficult. And I guess this is, again, another aspect that when you incorporate these biomarkers, then you're not looking at only at one biomarker, but you're looking at a collection of things. And you can come on back and say, patients that have this particular set of parameters tend to respond better. And I think that's very powerful because, first of all, you can look at it and say, like, maybe I can design, like when you mentioned like inclusion-exclusion criteria, I can design my clinical trials better so that I enrich for this particular patient. But also it feeds, it's lab in the loop. So it feeds your drug discovery portfolio and say, oh, these are the patients that are not responding to particular treatment. What are the things that I need to consider when I'm designing like a version 2 of my medicine to basically have a better outcome for this particular patient?
And when you say patients, do you mean the digital twin patients from
which
the
would
study?
be a replica of like your actual patients, right? So again, think of like the NHL population. You look at the data, you basically retrospectively look at the data and say, from like these 50,000 patients, about like 35,000 of them responded. That's great. What about the other 15,000? What are the parameters that were the bottleneck or the rate limiting for us? So we identify those and say like, okay, now if we want to design a new medicine, what are those parameters for that new medicine that need to change in order to be able to help those patients?
So your 15,000 digital twin patients that didn't respond, the way that the modeling works is the model will have changed those biomarkers in those patients too, based upon the input. And you basically look at those patients and their biomarker signature, which is a digital signature. You have not measured biomarkers in 15,000 people, but you believe that the variations you see in those biomarkers in the model reflect an amplification of what you would have seen in the 6 patients that you started with, or
that would create a hypothesis for us to test, right? So yes, exactly. So we are not measuring a lot of things in these patients. We are basically guessing or estimating, right, based on what we are measuring. Yeah. But when researchers or colleagues in oncology research, they are looking at like, okay, I want to design the next version of this molecule and I want to do it better, but I have 10 different parameters that I can go after. What are the things that I need to go after, right? That's when we come and say, when you look at these 10, if you change these 8 and make them like 10-fold better or 100-fold better, it doesn't impact the outcome. But if you change this and this, like if you change your potency in T-cell activation, for example, or your affinity to a specific thing, then you can have an impact.
And so you go in and you basically say, here, computer, here's a version of this drug that's slightly more potent and slightly better PK. Show me what this would look like in the patient set. And
Exactly.
you can see that.
So
That's
something
amazing.
that you would not be able to do, it's very costly. You don't, again, it's one of those scenarios that you cannot put like 10 different molecules in patients and see what happens. Those are the things that we do in the computer. We run the computer simulation and then we go back to our colleagues and say,
make this one.
Yes. Yeah. And then again, we refine.
A lot of iteration.
A lot of iteration, a lot of like lab in the loop type of experiment.
Yeah, that makes a lot of sense. That's very, very exciting. Have you seen anything surprising where you had a hypothesis and then you went back and you tested it and you're like, we were completely wrong. We're going to have to change the model.
We are in the drug development, so we get surprised all the time. Yeah. Some of the things that I've seen is we learn from a specific disease indication and we assume that whatever that we've learned in that is kind of like similar in a very disease indication that at least from like the measurement is similar to that. And then we go and test it in patients and the response is very different, right? So every time it's like very humbling to see that when you go with a new drug, even though the target is very similar, or when you go to another disease indication, even though it's similar to what you have tested, there are so much that we don't know. And the only way to actually get to know that is like to run a clinical trial, get that data and refine your model. It has happened so many times and it has led to so many different versions of our virtual digital twins. We have them for different disease indications, and as we get more data, we refine them, they get more differentiated.
That sounds wonderful, because the worst kind of surprise is something that fails in phase 3, right? So if you can predict whether that will happen or not before we even get into patients, that's going to really, hopefully, increase we use the term PTS, probability of technical success, of getting something to patients that actually works. So it's super exciting.
That's one of the hot areas of research that there are startup companies that work on basically creating this huge map of like different diseases, different cell types, different signaling pathways within the cell types. And the idea is a lot of drugs, they have efficacy. The efficacy is not 100%, it's like 30%, 25%. But there are patients that respond to these drugs. And the goal for sort of like this area is that we look at like 270 drugs that have been approved, and can we look at the combination of them and basically identify this niche of patient populations that would benefit from a particular combination. Something that has not been tested, something that is every single individual drug has been approved for, maybe we can combine them and essentially have more benefit for that particular patient population. So that's an active area of research.
So it's another way of getting at a sort of personalized medicine approach.
Correct. So the way that I'm looking at it is like, this is for the biotech industry and the pharma industry. Like, there is not enough money to test all the different particular combinations that you think would be relevant, right? So you have to be wise in the way that you pick them, and you have to use a lot of computational and statistical models to say, this is perhaps a combination that could help patients, right? So that's essentially the way that we are looking at it. It's like, you cannot just test all the combinations and say which one would work.
No, that's wonderful.
For those that don't know what a biomarker is, can you give examples of the types of biomarkers that are being used in some of these clinical studies and in these digital twin models?
So biomarkers are generally something that you can measure in a sampleable fluid, so for example blood, that is associated with the disease outcome. So an example could be blood eosinophil levels in asthma. So that's measuring a cell type. Or you might measure a protein, a specific protein. For example, you might measure a cytokine like IL-6, and does that associate with reduction in inflammation or something like that? And so what we do in clinical trials, we'll often always have a biomarker plan, and you will have dedicated biomarkers that you're measuring. But we also do exploratory biomarker analysis, and there are these sort of technologies where you can capture sort of 7,000 proteins at a time and assay all of them in a sort of fairly crude way. I mean, it would get kind of a high, medium, low level of the protein maybe you would get from that. And then that's what you can use those to do biomarker discovery. So for example, you've run your clinical trial, you have your outcome, you know who responded and didn't respond to the drug. If you collected one of these biomarker protein panels, you can then go back in and say, oh, okay, the people who responded to the drug had they also had an elevation of these 6 proteins or these 5 proteins, and then maybe you've identified perhaps a new signature for how a drug works, or to predict whether somebody might respond. So, what level of trust do you have in your models? How do we know that they're accurate?
It very much goes back to how much information or how much data you had to train the model on, right? If you have a very new treatment or a new target that we've not seen before, we've not tested it, other people have not tested it, there's nothing published, and you only have data from 10 patients, we would be able to predict things for you. But when we do the predictions, our predictions are going to be wide, and we're going to give you a range of things that may or may not be useful for decision making. So it really goes back to, if I'm particularly targeting a specific thing and we have 10 years of experience with that and we have 100 patients worth of data, we have way more confidence in what we are predicting. And the ranges that we have for our predictions would be narrow. So then we have an accuracy on top of that, and that accuracy basically comes back from the statistical pieces of your model. You have matched your data and then say, based on this data that I have observed, what would be the results? If your data is kind of from 10 patients, if you don't have a lot of knowledge, then your margins are going to be wider. So that's basically the way to look at it. There are times that you use one basically set of the data to inform the next thing. So for example, you go from preclinical data to phase 1. You don't need to be very accurate. You go from phase 1 to phase 2. Again, you need to be more accurate, but not, again, very accurate. But when you want to go from phase 2 to phase 3, that's when we want to be as accurate as possible. So the accuracy of the model also depends on the question that you want to answer and whether it's good for, or like whether it's fit for the question that you are trying to answer at that stage of the drug development.
What other limitations are there?
The limitations, I guess I would start from the fact that there's a lot of things that we are not measuring. So Obviously, like when you are running preclinical models, there is a lot more that you can measure. We can get tissue samples, all of those things. When you get to clinical trials and then you get to patients, a lot of those things we don't have access to. So basically your data becomes limited. But at the same time, because now it's in a clinical setting, things can be kind of not exactly as the protocol says. For example, you can say like, this is the dose level that the patient has to receive, and this is the day, and this is the exact time. The nurse that is doing that may not be sort of like doing that exactly at the same time because they are busy. So the timing would be a slightly different, the dose level may be slightly different. So all of those things are kind of like the limitations that you have to incorporate into your model. So I would say less data and also the variability in the data is kind of like the limitation that you have. The other thing is like, as we learn about different aspects of the drug, we may actually not measure those biomarkers anymore. Like for example, at the beginning you are very sort of like wide and say like, I want to look at everything that may be relevant to my drug. But as you get to know your drug better, then you also tend to reduce the biomarkers. So that basically reduces your data to some extent. So those are kind of the limitations that you have to deal with. But our models also at the same time are equipped to deal with those things. So I feel like there are limitations, but the field has improved and advanced to the point that we can deal with those limitations. The biggest thing is, at the end of the day, the number of patients in a clinical trial is going to be limited. You cannot have, let's say, for example, in an oncology indication, 5,000 patients. That's not happening. And that's, I guess, one of the biggest challenges for generating these virtual patients.
Your comment about the fact that we can't measure and don't measure as many things in later clinical trials is interesting because you've got more patients there, so more data, but less information about all of them. So what do you think? 5 to 10 years from now, how will we be using this in our industry?
So thinking about future, I'm very excited about like what we're going to see in the next 5 to 10 years. I guess there are areas that, particularly in the LLM and Gen AI, we are thinking of digital twins, but from a very different aspect, like basically looking at the medical history and looking at EMRs and other things, building a trajectory of a patient. So that's one space that is very active.
And I guess the power of genetics and genomics is only going to increase in the next 5 to 10 years too. So what do you think about the impact of those types of data on the digital twin models of the future?
There have been models in the industry that, particularly in the areas that we have more data, like think of like IO, and for the clinical trials that have incorporated like checkpoint inhibitors. We have database.
IO, you mean immuno-oncology?
Immuno-oncology, exactly. And then sort of like you have like databases that are like 4,000, 5,000 patients, and there is genomic information. So some people have incorporated those into the models with varying levels of success. I guess it's coming for other fields, like for example, for some of the cell therapy treatments, there is a lot of emphasis on measuring those omics data and then sort of like use that as sort of like we're looking at the outcome. But again, I would say like, because the space that you're looking at is multidimensional, particularly when you look at the genomic information, you need to have a lot more data to be able to have confidence to incorporate it into the model. So I guess that's an exciting kind of like space and direction. We just need to find the right treatment and the right indication that getting the omics data could essentially help us with the digital twins.
Maybe cardiovascular disease, where you're running 10,000 patient outcome trials.
Correct. So those are the places that essentially you can have a lot more impact and essentially be able to map the omics data to the system parameters of. So you can think of like 5 to 10 years from now, we have more mature models of different diseases and different indications that are of interest. So putting those things together, now you have a lot of powerful information that's coming from the vast information that you have from just hospitals and patients that are being treated, plus the models that are mature. And the combination of those can help you not only basically project what sort of like treatments would work, but what level of response you would get, and maybe perhaps even say, what is the prediction for this particular individual or this particular patient. I guess that's where we are going, and I'm excited about the future.
It's very exciting. Thank you so much for your time. I really appreciate this conversation. It's given me lots of things to think about, and I'm sure our audience too.
Thank you so much, and thanks for having me.
Great discussion, Maria. I'm curious if there is a world where digital twins could replace clinical trials.
I mean, that's a question for the FDA. right, who approve medicines. I think would be a long, long way in the future to have sufficient confidence to put a drug on the market without having tested it in a statistically powered way in patients. So I think it's pretty unlikely. I think it's more likely that what these digital twins types of tools will allow us to do is design better studies more efficiently, maybe fewer patients needed to be treated, maybe can accelerate trials faster because you're doing the right study the first time, right? That's where I really see the value. But I don't think we're going to see a world where you would replace a registrational package for a drug in patients with a digital package. You may see a supplementary piece of data to support expansion of a label into a different patient population, maybe, that type of thing, perhaps you might see. Or you already start to see computation being used to, for example, if you want to explore a different dosing regime of an approved medicine, perhaps you don't need to actually test all those doses. You can use some computational methodology to prove that would be safe based upon known data.
If you could use a digital twin for something, what hypothesis would you want to test?
Rather than a hypothesis, what I think I would anticipate this being really useful for is to generate data on a hypothetical very large patient population before you go into phase 3. So say, for example, cardiovascular outcomes, right? So one of the things that's really important for metabolic disease is long-term outcomes of our drugs. And those studies are really expensive, really long to enroll. And you're always taking a chance. You don't really know what's going to happen, right? So if there was a way using available patient data, public data, our data, to have a much better way to say, look, my drug lowers LDL by this much, it lowers hemoglobin A1C by this much, it's going to give you a weight reduction of this, and this is its basic molecular mechanism of action, it's affecting these enzymes, these pathways. Do you, digital AI twin person, what do you think is going to happen to the cardiovascular outcomes if I ran 10,000 patients with these parameters with this drug? That would be really amazing. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It will help new people to find us. And be sure to subscribe. If you have a question about the show, you can contact us at podcast@gene.com. That's GENE.com. And now for me, it's back to wrestling with data.